Welcome everyone to this workshop on In-Silico trials in regulation and funding decisions. My name is Mark Scolfer. I'm based at the Centre for Health Economics at the University of York. And this workshop is part of a grant from Innovate UK. We'll be hearing a little bit more about that shortly. We've used the term workshop, but I must explain the fact that we're not really in workshop mode. We have a number of people who are on our panel today as speakers and commentators, but we had a lot of interest in this activity, and we had over 100 people sign up, and we felt it really wasn't feasible to run it as a workshop. So we hope you'll bear with us in that, but we very much want your contributions as attendees. There is a Q&A button you'll see there, and we'll really welcome your comments and feedback and questions, and we'll leave plenty of time for that. And indeed, we will welcome feedback from you after the workshop as well. The other thing just to say is that we are recording this workshop. We're going to be using it so those who expressed an interest in the material we're covering but who can't make the slot can see it afterwards, and we'll also use it to develop some of our reporting as part of the grant, but we're not planning to publish this more generally. So I'm going to share with you just quickly the agenda for the meeting, for the workshop. We've got, so I'm going to ask actually just shortly Alex Frangi, who's the primary investigator for the grant, to say a little bit more about the grant and its broader objectives. But then we're going to move into the specifics of what we're focusing on today, which is working with in-silico methods, but also using decision analysis, value of information methods to support decision-making, both regulatory decision-making and what we're calling funding decision-making, which others might be more familiar with as health technology assessment. So Natalia Kunst is going to start by talking about the principles of those two different types of decision-making and the role of in vivo trial evidence. Then Marta Suarez is going to talk about how we can integrate in silico trial evidence in frameworks to support decision-making. After both of their talks, we will have a short opportunity for people just to ask clarifying questions, and you could do that as attendees through the Q&A facility, and our panel here can also do that. Then we're going to move into a structured panel discussion and Q&A. So we are hoping to have 3 commentators. We've got 2 of them on, I think, so far. Joyce Tate, I'm not sure whether she's yet able to join us. I think she was having some technical problems. Joyce is based at University of Edinburgh, who's going to talk a little bit about the regulatory side of things. Now, I should say that we haven't found it easy to identify. We would have liked somebody from MHRA in the UK to come and talk and give us their perspective. As some of you might know, we're in a pre-election period in the UK, and it wasn't easy for those colleagues to join us and to speak publicly. So Joyce has bags of experience in the regulatory area, and so she's going to talk on that. Hopefully she can join us. We then have Adalia Daywood from NICE, who's going to talk about perspectives of an organisation making funding decisions, HTA body. And then Parker Tyler, who's from Edwards Life Sciences, is going to give some comments from the perspective of a technology developer. Now, also on our panel, live on the workshop, as it were. In addition to Natalia, Marta, and the three commentators, Joyce, Natalia, and Parker, I've just mentioned, we've got Carl Claxton, who's another colleague at the University of York, who's been very much part of putting these ideas together that we'll be presenting shortly. And we've also got Mark Palmer, who's based at ANSYS, which is a US-based company which develops computer-aided engineering and multi-physics simulation software. And also Keith Abrahams, who's a biostatistician and data scientist with bags of experience in HTA and regulatory work based at the University of Warwick. So just before I pop to Alex for a little bit of an overview of the broader grant and research activity, in terms of objectives from the event, we're going to be considering, as I've said, analysis to support these two separable but undoubtedly very linked types of decisions. Regulatory decisions around the licensing and other regulatory frameworks for medical technologies and interventions, and funding decisions about the market access for those types of technologies, and decisions about their funding and levels of funding, levels of essentially price negotiation, price decisions. And we're going to introduce the principles of decision analysis and value of information analysis. So these are analytical tools that are used, or certainly decision analysis is used widely in some areas of HTA. Value of information analysis is also used, but to a lesser extent, to support funding decisions in HTA. And to some extent, decision analysis has been used around regulatory decisions as well. But we then want to understand how those methods can complement in-silico methods, particularly for the purpose of supporting decisions. So this workshop is very much focused on decision making and the extent to which in-silico, alongside a range of other potential benefits, can be alongside frameworks like decision analysis, VOI, can support decision making. And very importantly, we want to receive comments and feedback from those attending the workshop, both during but also after the event. So what I want to do now is just hand over to Alex Frangi. So Alex, based at University of Manchester, and I'm just going to briefly give us an introduction to the broader research in this area and this particular project. Alex, over to you.
Thanks a lot, Mark, and thanks for organizing this event. So I put in the chat some briefing information and some links that might be of interest, but basically this workshop is organized thanks to generous funding by Innovate UK through a particular program that I listed there. We are a community, it's a grassroots community of people that come from very different backgrounds, from academia, from industry, from standardization bodies, compliance testing, policy and regulations. We all believe that there are some limitations in current approaches and techniques, also a lot of benefits that have effectively guided the way we have done things so far. But we also think that there are limitations that could be overcome by complementing some of these existing techniques with additional techniques and go beyond sort of animal, bench, and human testing, adding modeling and simulation as an additional approach. And this funding that we received earlier this year is for what we call a discovery phase, or what Innovate UK call a discovery phase. And the main purpose of the discovery phase is to identify ways to build collaborations and partnerships, convening discussions like this one, hosting workshops like this event, engaging with communities that perhaps we haven't engaged before, and also prepare ultimately a submission for the implementation phase that will be in late August. And fundamentally, one of the communities that we identify already long ago through collaboration with Marta and Mark was, how do we articulate, how we formalize decision making in this area? Coming from engineering and computer science community, we were not at that point familiar with some of the approaches that Mark will talk about, and we think that they offer a nice blend between looking not only at the technology and optimizing that as an engineering design, but also look at the implications downstream in terms of potential cost, adoptions, reimbursement. And we hope that, that we will get a lot of insights from this, community, and also, get to know a number of people that might be interesting in reaching out and take things further. So I'm going to leave it there for Mark to continue, but, I want to really welcome everybody and, hope that this is an enjoyable event. Thank you.
Thanks, Alex. So for those of you who are attending in webinar format, very much encourage you to put your contributions on the Q&A as the speakers are delivering their talks. Don't feel you have to wait until the end. We can pick them up either as a point of clarification directly after their talks, or in the main sort of Q&A bit of the session. So we're going to start off with Natalia. So Natalia is a senior research fellow based at the University of York Centre for Health Economics, and she's going to talk about those key principles around regulatory and funding decisions.
Thank you, Mark. I'm just trying to share my screen. Can you see it, my presentation? Perfect, thank you. All right. So a big welcome from me as well to the webinar. And I will present a framework for considering evidence to inform regulatory and funding decisions, including decision analysis and value of information methods. So in this workshop, we'll be discussing two types of decisions, as Mark has already mentioned, regulatory and funding decisions. While regulatory bodies frequently make decisions on whether new medical interventions are effective and safe and should be marketed, funding bodies aim to determine whether the new intervention should be funded by assessing how well the new intervention works in relation to how much it costs the healthcare system. And both regulatory and funding bodies have similar principles for making their decisions. More specifically, they aim to provide patients with early access to effective and safe interventions. And to inform their approval decisions, they assess whether the benefits of the new medical intervention outweigh the associated negative consequences. When it comes to regulatory decisions, this assessment is based on benefits and harms associated with the new intervention. We estimate the incremental net clinical benefit, and if we obtain a positive value, the benefits of the new medical intervention are higher than the associated harms. And to perform this assessment, evidence from pivotal trials is usually required. When it comes to funding bodies, they also make their decisions based on incremental net benefit. But this estimate is positive if the incremental benefit exceeds the opportunity costs. And here, whether an effective intervention will improve health outcomes overall requires a comparison with the likely health opportunity costs. That is the improvement in health that would have been possible if any additional resources required had instead been made available for other healthcare activities. And these funding bodies aim to ensure that the decision to approve the new medical intervention is expected to improve rather than reduce health outcomes overall. And to determine this, we need several estimates. In addition to the benefits and harms associated with the new medical interventions that we used in our in the regulatory decisions. And these additional parameters, which we call here other, are needed to transform benefits and harms to long-term mortality and mobility outcomes. we express these health outcomes using a generic measure, such as a quality-adjusted life year. And it is important to use a generic measure because it enables comparisons across different diseases and populations. Further, we also need estimates on resource use and costs to derive opportunity costs. And these type of assessments that I'm describing here are mostly performed through health economic evaluations. Given the high number of inputs needed to estimate the incremental net benefit, a model is often used to inform funding decisions. More specifically, we develop a model that reflects the clinical practice. And this type of model is informed with a number of input parameters and predicts desired outcomes, which we use to estimate incremental benefit and opportunity cost, and finally, the incremental net benefit. And the large number of model inputs of various kinds, including clinical parameters, economic parameters, and health-related quality of life parameters, require gathering evidence from a range of sources. Depending on the input, the preferred study type of choice would be different. For example, while we would use an RCT to inform the input on clinical effect, we would prefer observational studies, which are not conducted in highly controlled conditions, to inform input parameters on disease characteristics, such as prevalence, incidence, or recurrence rates. And some model inputs may be currently unknown and cannot be informed using any evidence because no data exists. And in these cases, the unknown model inputs may sometimes be obtained through model calibration, which is a process of determining the parameter values that result in model outputs that fit to empirical data. In addition to calibration, other sophisticated estimation methods, such as statistical methods, may also be considered if the available data allow for indirect estimation of input parameters of interest. Importantly, the evidence base used to inform many of model inputs is often weak, leading to high uncertainty. So, as I have already mentioned, both regulatory and funding bodies aim to enable early patient access to new medical interventions, and they perform qualitative and/or quantitative assessments of the available evidence to inform their approval decisions. Here, they often face the question whether the available evidence is sufficient to make an immediate decision. When making this assessment, it is important to remember that there is uncertainty in every regulatory or funding decision because we only have access to a sample of the population of interest, not the whole population. And so the evidence provides a mean and precision estimate based on that sample. Consequently, there is always a chance that the decision the regulatory and funding bodies are about to make turns out to be wrong when more evidence is available. And what do I mean when I say that the decision may turn out to be wrong? Well, I mean that when we have more evidence, we may find out that the new intervention leads to negative incremental net benefits. Consequently, this evidential uncertainty needs to be considered when making regulatory and funding decisions. So for regulatory decisions, in the presence of evidential uncertainty, instead of using an estimate of benefits and harms, we use the distributions of clinical benefits and harms. And by subtracting harms from the benefits, we derive a distribution of incremental net clinical benefits as presented on this figure. And the part of the distribution where the incremental net clinical benefit is below zero represents the number of times that the benefits are lower than the harms, and this is our probability that the decision to approve the new medical intervention is wrong. For funding decisions, as we said, we have a number of model inputs. And instead of using one estimate for each of these inputs, we use a distribution that is informed by a mean and precision estimates based on the available evidence. We use these probability distributions for inputs in our model and rapidly calculate the model outcomes for many random draws. This way, we account for the evidential uncertainty. However, when we use a model to guide funding bodies in their choices, we need to also think about the structural uncertainty. When we develop a model, we aim to reflect the clinical practice. However, sometimes we may be unsure what assumption reflects best the clinical practice, because the evidence may support several scenarios as plausible. In this situation, it is important to examine the implications of different possible assumptions on model results. And there are several ways to account for structural uncertainty. One of the approaches is model averaging. So here we run the model with alternative structural assumptions and assign a weight to each assumption. In our example, we have two alternative assumptions, one defined as model F and the other defined as model G, as indicated in the figure to the left. We assume that both assumptions are equally plausible, and we weigh them 50% each to obtain weighted distribution of the incremental net benefit, as presented on the figure to the right. And after we have accounted for both evidential and structural uncertainty, we estimate the number of times incremental benefits are lower than the opportunity cost in our distribution. That is, the incremental net benefit is negative. And this represents the probability that the decision to fund the new medical intervention is wrong. So, as I have previously mentioned, there is always a probability that the decision we are about to make will turn out to be wrong. And as we have just discussed, there are methods to account for evidential and structural uncertainty and to assess this probability of making the wrong decision. However, the question is whether knowing this probability is enough to decide whether the current evidence is sufficient to approve the new medical intervention. So the probability of making the wrong decision is not enough to inform approval decisions. We need to also take into account the potential consequences if the decision we are about to make turns out to be wrong. That is, what would be the cost or foregone benefits? Here, we need to also think about the size of the population that will be affected by the decision, as the consequences of making the decision that will affect one or two people will be totally different than the consequences of a decision affecting several million of people. And including additional information to inform our decision is valuable because it reduces the expected negative consequences of current decision uncertainty. To assess whether collecting additional information would be worthwhile to improve the approval decision, we can use the so-called value of information analysis, which combines an evaluation of the probability of a wrong decision with the consequential loss function. So let's have a simple example of how this VOI, value of information method, can help us assess the expected consequences of making the decision based on the currently available evidence and determine the value of additional information to reduce the uncertainty in the approval decision. So in this example, the brown line indicates the probability distribution of incremental net benefit. We assume that the new medical intervention has a positive expected incremental net benefit. Consequently, the results indicate that we should approve this intervention. However, due to the decision uncertainty, there is a probability that this decision would be wrong and that we would need to face negative consequences. This is the red shaded area in the figure. If it turns out that the incremental net benefit is actually negative, the wrong decision would have been made, and the payoff would have been higher had the decision been to go with the standard of care instead of the new medical intervention. In other words, This is the loss, which can be expressed in health due to approving the suboptimal intervention. Now, suppose that we can include additional information collected through our research study, which will reduce uncertainty in the incremental net benefit, that is, reduce decision uncertainty. The results from this additional research study can be predicted with the likelihood function, where the most likely value of the sampled incremental net benefit is its prior value. And this additional data will tighten the distribution as presented with the black line. Thus, with additional information, the probability of making the wrong decision and the expected negative consequences of current decision uncertainty are reduced from that previous wider red shaded area to this small red shaded area. And the shaded green area is the value of reducing the expected loss associated with current decision uncertainty by including additional information to guide the decision making. So in this part, I have presented how decision analytic methods, including value of information analysis, can help regulatory and funding bodies make more informed decisions. As I have mentioned, there is always uncertainty in our decisions, and the value of that uncertainty is the value of further evidence that could be gathered to reduce the uncertainty. To answer the question whether additional evidence is required to support approval decision for the new technology, we need to answer questions such as how uncertain are the expected benefits? Does this uncertainty matter? That is, will it change the adoption decision? And how much does it matter? That is, what are the consequences of getting it wrong? And the decision analytic methods that I briefly presented here can help to answer these questions, and they can further help identify the type of additional evidence that would be the most valuable for the decision and the research design, identifying the research designs that would be worthwhile. And with this, I would like to thank you for your attention, and I'm happy to answer any clarification questions.
Thank you so much, Natalia. That's great. So opportunity now for points of clarification. Again, attendees, please do use the Q&A panel for that, but also colleagues on the panel, please use your digital hand to indicate you'd like to comment. We have had a question from one of our attendees, and I apologize if I'm mispronouncing your name. Midhan has asked what kind of model we're talking about here. So I think you talk quite generally in your early slides about the use of a model, and their question is about what type of model we're talking about.
That's a very good question. So we didn't want to go into too much into detail about that, as we wanted to just keep it pretty brief. But the models, because we were talking about models used for to inform funding decisions, those are decision analytic models. So mathematical models. There are different types as well, but those are more I would perhaps categorize mathematical versus statistical models. So those are the mathematical models that we talk about here, also called decision analytic models, such as Markov models, microsimulation models, and other types as well.
Carl.
Well, I mean, just to say that I think the principles are generalizable to any kind of model. And in fact, this is known as value of information within the HTA literature, but it has its origins way back in the late 1950s, basically in engineering, and has been used in engineering and risk assessment. So it applies to any model where we're trying to use the output of that model to inform a decision, and we care about whether that decision is uncertain, and we care about the consequences if we make a decision and we turn out to be wrong. So I think in general terms, you can substitute any kind of model in there. And I think what Marta's going to talk about is the fact that we can view In-Silico in a very similar way. It's a model, much more sophisticated, but not that dissimilar in principle to the kind of decision analytic models that are commonly used within HTA processes.
Thanks, Carl. Alex, we're just on brief clarification type points at the moment before we get into substantial discussion. Alex.
Yes, it's just to emphasize a bit what Carl started explaining, which is we have models for decision making, which is a thing what your community is bringing, and then there is detailed models on the performance of the medical products themselves. And I think the power here is combining these two approaches into something that brings them together. One that tells us how to understand the performance of the devices themselves or drugs. The other one is about how we combine the evidence that these provide into a proper decision making that is useful for regulations and reimbursement, etc.
Thank you, Mark.
Thank you for the presentation, Natalia. I wanted to ask a clarifying question. So it seems like the crux of the discussion rests on the B minus H term in this analysis. So when does the benefits outweigh the harm? And I was wondering if you could give kind of a broad brushstroke on what happens if we are able to reduce the harm side of this equation and shift the whole curve, like the ones that you showed in like slide 7, shift that whole curve more towards the right so that more of it is sitting on the benefit side.
Thank you, Mark. So just adding, before I answer that, just very briefly adding on the previous, that I totally agree as well with what Carl and Alex said. I think we can distinguish like model, as I was mentioning, decision analytic models, and then other types of models that we can use as inputs to produce evidence as an input parameters as well. And to answer your question, I will just lower my hand, Mark. That's a very good question. So where we are, as I was presenting, we are trying to as well make analogy between the two, or showing the similarities between the two types of decisions. But I think it's what I was trying to present as well, that there is uncertainty around both harms and benefits. And because of that, there is this chance as well that the decision will be wrong and there will be associated consequences. If we found out that with certainty there are no harms, but there are benefits, and we are sure, then we know that we don't need more evidence if there is a certainty. But then the point is as well, what is the evidence for that as well? How strong is the evidence, right? Based on how many, because as I mentioned, it's always based on a sample of the population, not the whole population. So I think it's this, as we were looking at the distribution, so how much is the negative benefit, like how certain we are about that? And if there are only benefits and there are no certainty, we are better off making the decision based on the currently available evidence. Although I think in practice that doesn't happen often, to be honest.
Thank you. Marta, is it on this point? Carry on.
Yeah, it's just to add that I think Natalia's presentation, what it highlights is that any activities, being research or development, that can contribute to discovering or finding out what the benefits and the harms are, or reduce any of the elements or increase any of the elements, can be, the value of them can be quantified by this kind of quantification approach that we use in funding decisions. So the decision analysis and value of information elements, they can quantify the value of these, not just research activities, but also development activities in improving the benefit harm profile of a particular technology.
Thanks. We've had a further question on the Q&A from, again, apologies if I'm mispronouncing names, Payman, who's asked who the stakeholders are in the harm benefit profile and how do you interject patients' preferences in the harm and benefit. Carl, you've got your hand up. Do you want to have a go at that one as well?
Yeah, for sure. So I think when it comes to the benefits of a new intervention, then we need to ensure we've captured all those long-term benefits, and bodies like NICE are very good at being very inclusive evidence to make sure we do that. In terms of how we measure those, then NICE tends to use generic measures which are based on patient preferences in terms of trade-offs between different dimensions of health and also survival. So, however, on the harm side, as well as potential harms that are borne by the patients who also benefit, we also need to take account of the additional healthcare costs and where they fall and what the consequences of those costs are. So that's what Natalia was talking about when she mentioned health opportunity costs, that if there are additional costs, those costs are going to have to come from somewhere else in the NHS. They're going to displace care that could have benefited other patients. And we now know something empirically about what that looks like. So in the context of funding decisions, it's predominantly about benefits on one side versus harms in the form of opportunity costs on the other. Now, there are different ways in which we might measure health benefit, but by and large, we attempt to try and capture patient preferences as we do that.
Thanks, Carl. Before I come to you, Keith, Alex Thompson's posted a question on the Q&A about what types of interventions we are thinking about and whose perspective we are adopting. So is it the developer or the regulatory reimburser? So if I can just quickly air a view on this myself. So interventions here, in principle, we're talking about any intervention, any medical intervention, technology, otherwise. I mean, I guess primarily we're talking about proprietary interventions, technologies, those with a commercial sponsor, but not exclusively. The perspective, well, the focus today is the decision-making context. So the decision-maker's perspective. So from a regulatory perspective, that would be in the UK, for example, MHRA, European Medicines Agency, and FDA in the US. But also those organizations making funding decisions, which are numerous, but would include, for example, so-called HTA bodies like NICE. Clearly, in those decision-making contexts, the perspective those analyses take is in part a discussion. Certainly in the HTA field, that is always a point of contention. It's certainly for NICE, it'll be the benefit, health-related benefits to patients and others, together with costs falling on the National Health Service. But there is some variation around that internationally. Keith, I presume you're not on that question, but on an earlier point?
No, it was really about harms. In that I thought it's perhaps worth reiterating the point that often we have more evidence on the benefits, or at least potential benefits, whereas for harms, even though from a regulatory perspective, that's perhaps what they're more interested in, or at least it has prominence. We do, there are numerous examples where in the UK, for instance, NICE has made a decision on a drug that then has been shown to have rare but potentially, sort of significant harms, and has been withdrawn from market. So it's perhaps just worth thinking, remembering this fact that we, there might be uncertainty on the benefits, but actually there might be quite a lot of uncertainty on the harms as well.
Thank you, Keith. We've had a question from Giovanni, what I'm going to suggest, and I was just typing to this effect, that we leave that question, come back to it after Marta's presentation. because I think it will help address that question. So Marta, over to you now to talk about the framework in more detail, how it can work alongside in-silico methods. So if you want to share your screen, Marta's a professor, again, based at the Centre for Health Economics, University of York. And I think probably done more thinking about in-silico methods from the health economics perspective than most.
Okay, thank you, Mark. Can you see my screen, the presentation element? Okay, thank you. So what I'm going to talk to you following Natalia's session is how this decision analysis and value of information framework can be extended to appropriately and efficiently consider the use of in-silico trial evidence in support of decision making. So I guess just to frame the kind of context of this presentation, and knowing that modeling and simulation is used and can provide support for many of the elements that are looked at in regulation and even in funding processes, we are here really grounding this presentation in silico trials, abbreviated here to ISTs. So these are kind of defined here by myself, so potentially not too accurate, but these are defined as experiments that evaluate comparative efficacy, effectiveness of an innovation, but that are performed partially or entirely using individualized computer simulations. So in this presentation here, I'll focus on the case where IST, the in-silico trial, informs the assessments of clinical value, so the benefit-harm profile or net benefit of the technology. And this is really akin to the current role of randomized controlled trials, RCTs, currently have in regulation, and then in supporting evidence in funding for funding decisions. So I guess just one note that I guess for just the sake of definition, I guess for in silico trials, it's worth noting that causality, which means the comparative effect of the technology in relation to a comparative effect, is determined, I'm here considering that is determined by modeling and simulation. That is, it's grounded on a mechanistic relationship and considerations, a bit like a model, as Natalia kind of grounded her presentation on. So the causality in kind of RCT evidence is typically achieved by randomization. We'll get to that in a couple of slides. So because we have quite a varied audience, and there might be people not so familiar with in silico trials in the audience, I here present an example which Alex here in the call has published. And you can see here the print screen of the paper in case you want to refer to it later and have a look at an example of an IST, an in silico trial. So this trial, just very briefly, is an in silico clinical trial that assesses the performance of flow diverters in the treatment of intracranial aneurysms. So in this trial, imaging data, I believe, was used to, by modeling and simulation, reproduce 82 distinct anatomies of intracranial aneurysms. And then the computer modeling used computational fluid dynamics to quantify kind of flow reduction with and without flow diverting stent in the same anatomy. So the results of this study, it's interesting to have a look at the paper, were shown to replicate the results of 3 in-human RCTs. So as I've mentioned at the beginning, but just to extend a little bit, in this presentation, I aim to extend the decision analysis and value information framework to consider how in silico trials can support regulatory and funding decisions. And I aim to do so in a way that appropriately acknowledges how in silico trial evidence compares to in vivo evidence. And here I'm referring to RCT evidence when I mention in vivo evidence. So going on to that point and kind of grounding the presentation and kind of the considerations of in-silico versus RCT evidence, in-silico trial versus RCT evidence. So as I've mentioned previously, the RCT evidence that typically supports kind of regulation and funding decisions is expected if relevant and well-designed studies are made available, is expected to provide an unbiased estimate of clinical value of that incremental benefit that we're here assuming is our metric that supports decision-making. So we're considering the case here where the in-silico trial provides evidence and provides an estimate of the clinical value as well. And being a model, in-silico and in-silico trial being model-based, it will include not just parameter, but also the structural uncertainties that Natalia mentions, and in a way that kind of aligns very neatly with decision analysis, with the framework that Natalia presented. So I guess the key consideration here is that contrarily to a well-designed RCT study, the IST estimate of incremental benefits is not guaranteed to be unbiased. So this estimate is only unbiased if all structural and parameter uncertainties are comprehensively and appropriately characterized, but we cannot ensure that. So the implications of this is that, sorry, there's been a bit of misformatting in the slide, but the implications of that are that with RCT evidence, we can gauge bias of the IST estimate, but without RCT evidence, we should really be considering model credibility when we think about using ISTs to support decision-making. So again, just trying to merge the two kind of areas, the in-silico research with health economics, funding and regulation science. The term credibility is really is a term that actually aligns quite well, is a term used in the in-silico trial modeling and simulation world. And it refers to a framework developed by the FDA to assess model credibility, but it aligns quite well with the principles of decision analysis. And this slide aims to show how well it aligns. So in the FDA's framework, there is grounded on two key concepts, the concept of model risk and the concept of decision consequence. So the concept of model risk is defined as the possibility that the model may lead to a false incorrect conclusion about device performance, resulting in adverse outcomes. So that aligns very well with the concept of probability of error that Natalia presented. The second kind of concept on the FDA's framework for model credibility is the concept of decision consequence, which is defined as significance of an adverse outcome resulting from an incorrect decision. So that concept is really equal to the concept of consequences of error that, again, Natalia referred to and is the basis for value of information analysis. So this slide just demonstrates how well integrated are the principles of decision analysis and value of information with how in-silico trials have been considered in policy. So on the core of the presentation, so how can IST evidence be used in decision-making? So without RCT evidence, I guess a decision maker will have three options when faced with an in-silico clinical trial to support their decision-making. So the first option is to assume that the IST, the in-silico evidence, has no credibility. And so to not consider at all the IST evidence in their decision-making. So the distribution that will be used to describe the existing evidence on the clinical value of this technology is not going to be influenced by the in-silico trial. So it's likely to be using other evidence and likely to be very vague or uninformative in the absence of other evidence. So if you kind of if you're coming from a statistical background, you'll see that the use of the term prior refers to Bayesian statistics, and Bayesian statistics really is a useful framework to use here because it allows us to make direct probability statements about particular parameters. So we can describe a parameter like the incremental benefit as a distribution and directly refer to that. And that aligns very well also with the concepts of probability of error and model risk that we've been referring to both in the in-silico kind of world, but also in the decision analysis world. So when using such a vague and informative prior, the decision maker, being regulator or funding decision maker, is likely to find that the value of the technology is very uncertain, depending obviously on costs, and that the value of information, if quantified, is likely to support the need for further research. So the second option the decision maker has is to assume full credibility of the in-silico trial and use the in-silico evidence to specify the prior that's going to be considered in decision making. So that is to describe the value of the technology and uncertainty over that. So if using the IST and assuming the IST adds to the body of evidence, so getting some precision as in the figure here, so you can see more precise distribution than the previous one, then you would say that potentially the value of the technology is less uncertain, always depending on cost, and the value of information is less likely to support further research. So the third option is to in some way weigh the in-silico trial evidence according to the credibility of the model used to define the in-silico trial. So this would allow assume a probability, 0, 1, or somewhere in between, a probability that the in-silico trial is credible. And this would allow us to form a mixture of this vague prior and of the in-silico estimate to inform the decision-making process. So you can see here in the graph in gray, the very vague prior. I'm hoping you can see my cursor. You can see the in-silico estimate in gray as well. And you can see the weighted mixture by assuming a probability weight of 0.2 for the IST, 0.5 or 0.8. So by increasingly weighting the IST evidence in relation to the vague prior, you can produce different descriptions of your uncertainty over your clinical value. So in this way, you can moderate the influence of the in-silico trial informing that description of the value and uncertainty over value of your technology. So when RCT evidence becomes available, then you can update that prior distribution with observations on your unbiased incremental benefits that were generated by your sampled RCT evidence. To form kind of your decision-making posterior. So again, Bayesian terms, prior and posterior. So prior, the data updates the prior to form the posterior distribution, which describes the joining of both the data and the prior. So you can see here a prior that is equivalent to one that we've seen in the previous slide with a weight of 0.8. So it's weighting the IST at 80%. You can see in blue a description of your true value. So this is obviously, I'm produced these curves assuming that the true value is the blue vertical line. And if you notice, the IST estimate is purposefully inconsistent with that true value. So just to show you how updating with data would allow correcting for the bias of the IST in this case. So if you have a sample size of 5, in the case where you have an inconsistent IST estimate with your true value, with a sample size of 5, you can see that this gray distribution is looking more imprecise. So and it's more imprecise because of the inconsistency between the data and the prior. So it's losing a bit of precision. As you increase the sample size, the data dominates the prior, and you're starting to gain precision and correcting for bias quite significantly. So you can also use the RCT evidence to update the credibility weight. So in this case, where you had a conflict, at some point, your credibility weight would be lower and lower as you're updating. Your data is showing that your credibility is actually not as high as you expected. So by updating the prior, you will be using, as you accumulate data, you'll be using more and more of the data and less of your IST prior. So in this context, so with this framework, the use of RCT data is actually made, or the use of RCT data is very efficient. And that's because any additional sample that was required to disprove your IST is used efficiently after the IST is disproved. So you kind of use data very efficiently. I'm going to try and communicate that in a slightly different way. So in this graph here, what you have is the same kind of truth, the same sample, so the same level of data, but different initial IST estimates, which I'm not showing. So I'm just showing the final estimates. So in green, you have your the case where you only consider in vivo, the RCT evidence. So that's your green curve. If your IST used to inform your prior is consistent with your in the RCT evidence, then the weight of that credibility weight of that IST will be updated to be a very high weight. So you're going to gain precision in relation to using the in vivo data by itself. If your IST is inconsistent, but the data is sufficient to disprove that, then your final posterior using the same sample size is going to reflect the exact same distribution as if you only had your in vivo data. If your IST is inconsistent, but the evidence collected wasn't yet sufficient to fully disprove the In-Silico trial, then you might generate a more imprecise estimate, so lose some of the in vivo evidence. But that as you collect more evidence, that will be resolved. So just as concluding remarks, so I think decision analysis and value of information can do a number of things. Can quantify the value of the use of the technology, can quantify the value of evidence to avoid consequences of decision error, can align with existing credibility frameworks and assessments of model credibility to ground the weighting of IST evidence in relation to RCT evidence. This framework can also formalize decisions about regulation and funding of a technology and link these to requests for further evidence in a way that is efficient and appropriate. It can, as a last point, it can support, and I know that this was a question we've kind of touched upon already, it can support not only regulatory and funding decisions, but it can be used to optimize R&D and investment decisions. And I think I'm going to terminate here. Thank you so much for listening in.
Thank you, Marta. It's excellent. As before, we'll have relatively brief, and I'll try and make it a little briefer as chair this time. Points of clarification. I'll come back to Giovanni's question on the Q&A from earlier at the moment. But Carl, I think you were going to just say something quickly about innovation in this context.
Yeah, we we've deliberately not said much in this presentation about the need to support innovation, but I think, well, I hope it's clear that this kind of quantitative analysis is not a substitute for decision making and the judgments required at all. Rather, it does make those necessary judgments and decisions accountable to reason, evidence, and explicitly stated social values. And so it means we can also address the need to support innovation. You see, at the heart of this is trying to estimate the long-term value of a new innovation and how uncertain we might be about that. Now, we actually know quite a lot. We've got good evidence about how reward or payment, the quantity and quality of innovation responds to payment. And so it means that we can also use this framework to identify the most appropriate reward or share of value that should be given to the innovator, mindful of the implications that has for future innovations, both in terms of quantity and quality. So I think we have an opportunity to explicitly address those potentially conflicting objectives of supporting innovation, ensuring that the scale of benefits justify harm, or whether the innovation offers sufficient value for money. So it's not something that we've incorporated in the presentation, but that question of supporting innovation can be integrated in the same quantitative framework.
Thank you, Carl. That's helpful. And that to some extent addresses Alex Thompson's point from earlier, which is actually still open on the Q&A, about the different perspectives here. Mark, have you got a point of clarification you'd like to make?
Well, I guess it's a question that may add to more confusion. So it seems like the underlying premise is that the RCT is the ground truth and is the authoritative representation of reality. What happens to this framework if the RCT is also treated as a model on equal footing with other sources of evidence, and that the ground truth is still unknown?
Good point. Marta.
I think that's the question we're all have been debating, all of us, with real world evidence, single arm trials, is how we're going to obtain that comparative and biased estimate that we so need to support decision making. I don't have an answer to your question, Mark.
Well, I suspect we might come back to that. I can see Carl poised to come in, but I'm going to leave him out for the moment. We'll come back to that because it's not a brief consideration. Let's quickly, Marcia, if you wouldn't mind, just quickly think about Giovanni's question from earlier, which I think you've addressed, but maybe you can say more, which is it possible to ask for better quality data along with more sophisticated methods? Or how do we make sure that the results are driven by evidence and not by structural uncertainty? Anything you can help with there?
So structural uncertainty, I think, is just a source of uncertainty as any other. I don't know if others would agree with me. I think the key consideration is how we ensure that any judgment on credibility of evidence is made in a way that is consistent with the principles of decision making. And I do think, for my reading of the kind of model credibility framework from the FDA, it feels that the principles of assessment of model credibility are very well aligned already with the decision making context. And therefore, I think we can consider model credibility appropriate in trying to reflect structural uncertainty and the quality of the representations of structural uncertainty and parameter uncertainty in silico trial evidence.
Thanks. Again, we may come back to that. What I'd like to do now is to move over to our three commentators, as we've called them. And so representing slightly different perspectives, just to give us a short commentary on how they view this broadly defined, this framework and these methods. From firstly, a sort of regulatory perspective, secondly, a funder's perspective, and thirdly, from a technology developer, manufacturer perspective. So we're going to start with Joyce Tate. So I'm glad you can join us now. We had a few technical problems earlier. Joyce is based at the University of Edinburgh, and perhaps, Joyce, if you want to say more about yourself, please do. But you're just going to give us a short perspective from a regulatory viewpoint.
Yes. So I'm Joyce Tate, based in the Inogen Institute in the University of Edinburgh. For the last 4 years, I've been a member of the Regulatory Horizons Council, which was a completely new body when it was set up. It was set up with the government's intentions to develop more pro-innovation regulatory systems for all kinds of technologies. And we've been doing work since about 2002 and a bit earlier than that on the number of times when a regulatory system was inappropriate to the needs of 21st century technology and actually had the effect of stopping innovation from happening in quite a few seriously important areas. And the solution to that was to make the regulatory systems more proportionate to the needs of the technology and more adaptive compared to the previous regulatory systems that were in place. And not only did we do that, but we were saying, how could you do it? And we've developed an approach called PAGET, Proportionate Adaptive Governance of Innovative Technologies, which explains all the things you need to take account of in a systemic way and how you bring them together to deliver what you need. And it brings in things like technology readiness levels. One of the other things it brings up is the creative use of standards to help to adapt regulatory systems more rapidly and more creatively and more supportively for new technologies. And various other aspects, I won't go into that in too much detail, but that particular piece of research was behind the setting up of the Regulatory Horizons Council. And we've used it as, I'm a member of the Regulatory Horizons Council, we've used it as members of the Regulatory Horizons Council in a few of the reports that we've done. So we developed it in the life science area mainly, looking at everything from gene editing and GM crops through to stem cell therapies, drugs, diagnostics, things like that. So we've got case study examples of what went wrong, how they could be adapted. And also we've extended that through the Regulatory Horizons Council into one of our reports on neurotechnologies, a report on hydrogen and transport, most recently a report on quantum technologies, where the quantum developers in the industry looked at what we'd done with this PAGET report and said, that's what we need for quantum. And it was really gratifying from my point of view to see them saying, this could also work with quantum. So I think it's quite a widely applicable approach to these kinds of things. Now, bringing that up to what you guys have been talking about this afternoon about the Keirsey approach, and I think, yes, it's really important, the kinds of things you've been talking about that could be done here with clinical trial systems and adaptation of clinical trials. And I think one of the things I would emphasize about that is that there's always an assumption that when you change something in the health service, regulatory or not, it's more expensive than it would have been before. And I think it's really important to emphasize with this kind of thing is that it could be cheaper and faster. Maybe not immediately, but it could lead to something that was very quickly cheaper and faster than the way we do things at the moment. It could enable you to get things onto the market faster with less effort and time in the clinical trial stages, and therefore with less cost to the developer at the end of the day, so cheaper drugs and cheaper treatments and so on. I think it's really important to be able to emphasize that and to start collecting some evidence around how that could be the case in future. And I also think, and this is something I'd love to talk to with others on the project, I think there are other ways in which it could become more useful and more important in addition to the ones we're talking about today. I think if you started using this kind of CST the approach, not the randomized clinical trials, but the the ISTs. And if you start using that earlier, you can speed things up even faster. I'm thinking about what happened in the COVID trials for the vaccines. As I understand it, what they did there was they did stage 2 and stage 3 clinical trials in parallel. So they were doing them both at once. And normally they do stage 2 first and then stage 3 in order to help save money. If stage 2 doesn't work, you've not spent the money on stage 3. Now, I think what the government did was took the risk that stage 2 would be okay, and if they'd already done stage 3, they would get the stuff on the market faster. That's how I understand the process that they set up. And that meant that it was developed very much faster. Now, I can imagine that in-silico trials could be a very useful aspect of making that a greater certainty than it was when we did it for COVID. That it would be, you would be in a better place to bet on doing phase 2 and phase 3 clinical trials in parallel rather than in sequence. And I think there are other things like that, other areas where if we sat down and thought about it, there are a lot of areas where the new agenda that the government in Westminster just now has is to really strongly support pro-innovation regulatory systems across the board for all technologies, including AI, quantum, drugs development, all sorts of things like that. It's across the board. It's an agenda for the current government. And as far as I understand it from my connections in the Regulatory Horizons Council, the potentially new government, if it's a Labour government, has also committed to beefing up the Regulatory Horizons Council, to supporting this agenda and giving it more emphasis than has been the case in the past. So I think it's here to stay, at least for another 5 years. I think it's something that this project and in-silico trials could really have a transformative effect on whole industry sectors. And I think we should be looking through this project to actually be able to say a lot more about the different ways in which it could be beneficial and the number of different ways in which it could actually make things faster and cheaper, and therefore make the drugs at the end of the day cheaper and easier to apply. I'm happy to talk about any of these things in more detail, but that's what I'd like to see happen as a result of this project. Yeah.
Brilliant. Thank you so much, Joyce. As you say, I think we'll be coming back to some of those issues a bit later. So we're going to go next to Dalia Dawood from National Institute for Health and Care Excellence, who, as you know, as most people will know, I think, is a key decision maker around funding for the UK or English and Welsh NHS. Dalia, over to you for some comments.
Thank you so much, Mark, and thanks for the invitation to contribute. So let me first start by clarifying that I'm going to be providing a general HTA methods research perspective rather than NICE decision-making specifically, and views expressed are my own. I also will primarily reflect on our experience of working or looking into modeling and simulation data in my team's work, which is our research, the funded research projects, and our newly established function, which is the Health Technology Assessment Innovation Laboratory, or HTA Lab, which is the current sandbox environment that NICE established to look at potential solutions to complex technologies that are likely to come our way and prepare for the future assessment of these technologies. But hopefully there will be some kind of reflections also on the potential use further down the line in actual decision making. So I think we are perhaps here with modeling and simulation data where we were with real world data and real world evidence around 5 years ago. And we are slightly ahead with these types of data than we are with AI and machine learning methods, given that there is some experience, at least from the regulatory side, in terms of using and considering modeling and simulation data. So we sometimes HTA starts to consider some innovation in terms of the methods specifically when regulators actually start looking at these types of innovation and start using them in making their own decisions. And I would draw the parallel that we heard from Marta about single-arm trials and external control arms. So these were used by regulators, and then HTA agencies had to start thinking about how they can use them for decision-making. So we are trying to avoid that situation by being early involved in this space. And a key part of our work in the HTA Innovation Laboratory is really thinking about these approaches. And so we have been exposed to modeling and simulation data use in one of our research projects, which is ERA for TB, or this project is a European-funded project that looks at the development of medicines for drug-resistant TB or tuberculosis, and developing a whole pipeline of innovation. And modeling and simulation plays a key role in this project in informing the design of RCTs, and as we mentioned, improving the efficiency of the whole process of drug development. So definitely reducing costs and reducing the reliance on using animals, for example, in testing. So at the very early stages before you start in vivo, but also simulating the potential outcomes from putting these drugs into RCTs. So we can see the potential for these data from an improvement in terms of efficiency of RCT data, or the conduct of RCTs in general, improving their efficiency, reducing costs, and speeding up innovation. But generally, I think using these innovative types or different types of data compared to RCTs, like what we are considering today, again, hasn't been or isn't new for NICE. So NICE started looking or used in vitro data during the COVID, as Joyce referred to. Some of the issues that we have learned to live with perhaps during the COVID times is the absence of RCT data and the need for making quick decisions, but still rigorous and high quality decisions using all the available data. So in vitro data were considered in decision making at that time. We also saw some attempts also for using susceptibility data. And I guess, Mark, you've been involved in that, developing antimicrobial evaluation framework. So again, in vitro data was used in this regard. Modeling and simulation data is a step ahead, perhaps in the development pipeline, and where we have slightly more information to draw on. So theoretically, you can think that you have more information to draw on and you're able to have better confidence in that live at this stage of development when you have modeling and simulation data. So I'd say this is still very early days, but in using such data by HTA agencies, and just very experimental for us at this stage, trying to look at the prospect of using modeling and simulation data. And at the HTA Innovation Laboratory, we are interested in the topic and potentially considering further exploration of how this data can be helpful for HTA decision-making. But general reflections, I guess, for such a framework to be useful, generally building confidence in the data that you're using. So external validation of their use against trial data would be very helpful. Marta put an example of a study that looked at external validation versus trial results. Second to what, again, I'm going to draw the parallel with real-world data, where we had started to become more confident when we saw studies like duplicating or replicating RCT results using real-world evidence. Having more development in terms of the actual methods that are used and being confident about the methodological rigor of using them is also very valuable. I think with modeling and simulation data also, it's useful to try and develop even further to link the results of these simulations to pharmacoeconomic modeling or economic modeling, so that you have this whole pipeline or linked analysis where you use the data and link it to economic modeling that could help make it even more tangible how you can use it in decision making. And we have seen it before with BKBD data or pharmacokinetic and pharmacodynamic data. There have been attempts to using these types of data and link them to pharmacoeconomic modeling, so that you have estimates actually of value or the cost effectiveness. And these have been used in informing go-no-go decisions by companies, for example, internally. They haven't been picked up that much within the actual decision-making context but it is very useful, again, parallel, to look at how we can use different types of data to address uncertainty or to quantify and characterize uncertainty. In general, having these data to help us with characterizing and quantifying uncertainty and providing further insight will be very useful to support decision making, but we need to develop more confidence in the actual value and quality of the data and the outputs from them. But operational factors, I think further down the line from a decision-making perspective, we really need to start understand how this can influence the needs to upskill perhaps the HTA world to understand these types of data and to work with it. So there are many operational factors to take into account when we think about any type of innovation that we are considering at the moment within the HTA lab, is how to implement it actually in practice, where we require changes in processes, upskilling of analysts, new committee members with backgrounds and expertise in these new areas, external assessment groups needing to upskill to understand these types of data and how they can work with them and their limitations. So I think that will be eventually the kind of the operational factors that will need to be considered. I'll stop here and perhaps we can explore a bit more once we hear the industry perspective as well.
Thank you very much, Dalia. That's great. So for the third of our three commentators, going to go to Parker Tyler. Parker's based at Edwards Life Sciences. Going to give us a broad perspective on how this might land with that sector. Over to you, Tyler.
Yeah.
Parker, sorry.
No, you're good. Thanks for inviting me. It's an honor to be here. And amazing sessions. It's really exciting to hear everything that you guys have to say. I'm very new to this world. I'm very excited about this world. It really comes down to one thing for me, and I'm going to explain that. But first, I want to ground the group. When it comes to innovation, I think of innovation as the product of invention and commercialization. And when we think about the rate of being able to innovate, it is dependent on the rate in which you can learn, right? And the higher the rate you can learn, the faster you can innovate. I think today we are very constrained, or that rate of learning is very constrained by the physical world, right? So from ideation to product development to commercialization to post-market surveillance and really understanding how products are doing in the field, it is still constrained by that physical world. And so where I'm really excited and curious about In-Silico and the potential of In-Silico, it comes down to how can we increase our rate of learning using the digital world to have less surprises in the physical world, right? And if we really understand that question and get deep with that question, and I think this is agnostic to many areas in industry, we are going to be able to get products to patients that are in desperate need of these innovations at speeds that are just beyond what we can do today. And that's really my motivation of why I want to learn more about this world, why I'm trying to get more connected in this world. And then, the challenges of adoption in industry, I think it's classic change management, right? Whenever you have this transformational technology, these transformational ideas, it is hard to adopt those transformational ideas in the existing minds that are making the decisions. And so when you think of change management, and I've seen this throughout my career in engineering, there's no shortage of good ideas and technology. The bottleneck is always the change management and the adoption. And so how can we increase that change management and be really strong at change management when it comes to this transformational technology? And to do that, in my experience, is you have to show it, right? You have to prove it. And so examples and so I'm very excited with working and learning more from Alex is, hey, give me some examples. Show me where this has landed, how it has worked, what it has saved, what it's done. And that is going to be fuel to drive the change in this world. So I think, the opportunity is insane in every possible way when it comes to innovation and the quality of products that we're delivering to these patients that need these innovations so badly. But then, how do we do that? And how do we come together as a community to start to drive that change. So I don't know if that's helpful. I just asked maybe more questions than I gave answers, but it's something I'm very curious about learning more and helping to do that.
That was very helpful, Parker. Thank you very much indeed. And again, I'm sure we'll come back to some of those points. So encourage, again, attendees to put any thoughts or questions on the Q&A. We have got a couple to come to, but I'll also ask the panel to chip in as well. So Thomas Walker, well, I think is also the Thomas Walker at NICE, has put a question about whether there are tools to help assess the quality of ISTs, to help with this credibility assessment and weighting. So I think that's an important question about, how are these weights established? Is there anything that we can clarify on that? So, I think there's two perspectives, and perhaps we'll start with Alex, who can give us some view on how that's been done in the IST literature to date. Alex.
Yeah, actually, it's really to seek Mark as well, who has been involved in this area to contribute. But I just want to say, I put on the chat another of the workshops that we are organizing that is precisely dedicated to answer that question. So that will be a 3-day event virtually, where we have engaged with colleagues from FDA and industry that are doing this, and we will walk through specific examples. But there is already some guidance from FDA in terms of credibility assessment, and we are working basically on taking them further. One question I want to leave there for Dalia, if she could help us there, is how we disentangle. I love the comment you made about how we are in relation to real world data, and I have had similar conversations with colleagues in MHRA that are essentially saying exactly the same thing. And we are working in taking the current guidance on real world data and look at how we can transform that into a guidance that we think will have two sections. One's on the quality of the data that fits the model, and the other one on the credibility of the models themselves, which is what we are going to address in this workshop that I put in the chat. But it would be interesting to know how we disentangle these two elements, right? The data that fits models from the credibility of the models itself. So the uncertainty on, let's say, the measurement uncertainty, if you wish, and the quality of data uncertainty with uncertainty, the epistemic uncertainty that we have on are the models taking into account everything we would want to take in the first place.
Thank you, Alex. Mark, do you want to chip in?
Yeah, and if I may, I wanted to add some specificity to some of what Alex said. So one of the challenges from I'll put on my old hat in the medical device industry, one of the challenges that we had there with modeling and simulation versus physical test is if you think about the questions that are asked of a physical test, we were asked to report the variability in the data, but not the uncertainty in the measurements. And as soon as you use the same data in a simulation or in a in-silico clinical trial in a computational model, you're asked to account for not just the variability, but also the uncertainty, and to track the uncertainty all the way through the simulation. So even though we had these sophisticated models that we could use internally, the burden of reporting all of that uncertainty externally made it far easier to just report the experimental data or the physical world data than to report the model, because so much was being asked of the model that wasn't being asked of the experiment. So that's one of the things that we wanted to think about. But then when we look at the primary questions around in silico clinical trials versus RCTs, a lot of the questions go back to this question of harm and benefit. And one of the dirty secrets is that we have a huge issue with reporting adverse events in clinical settings and in clinical trials. And if you look at the literature on adverse events, the optimistic ones say that we're capturing about 44% of the adverse events. The pessimistic ones say that we're between 5 to 10% of capturing adverse events in pharma and less than 1% of capturing adverse events in medical devices. And the numbers are even far lower if you're capturing the events with enough information to do a forensic analysis. So the underpinnings of benefit versus harm, the underpinnings of these clinical studies where one of the primary outcomes being tracked is morbidity and mortality, it's on shaky ground. I mean, we don't have reliable capture of the harm side.
That's really interesting. Thank you. Keith, it's similar sort of area. I don't know whether you also want to reflect on Giovanni's question here, which is sort of related, which is also about sort of some of the transparency challenges here.
Yeah, the point I was going to make. Well, actually, there were 2 points. What about the parallels with real world evidence, and the same transparency issues abound there. And you know, I think Dahlia's point is correct, and you know, the fact that we could look at the real world evidence, what's happened with real world evidence, maybe there are some learnings from that. But one thing, and this links back to Marta's presentation, is that when we see discrepancy in the situation where we might have RCT evidence, maybe limited, and we see discrepancy with in-silico trials, I think one thing is to take that discrepancy into account, but the other is to try and understand why we're getting a discrepancy. And I'll give an example from the real world evidence field, which perhaps illustrates what I'm talking about, is that, you know, we might see that we've got a different answer in our real world evidence to what we've got in the trials. But when we look at the populations, we see that in the real world evidence that there are there's differences in the population, and that might explain some of it. So I do think there are potential learnings. The second point I was going to make, which is similar to the parallels with real world evidence, is that you know, people are talking an awful lot about synthetic data, and what we're talking about in terms of in silico, there are parallels with synthetic data, and again, people are sort of you know, there are obviously arguments about that we should use synthetic data for you know, helping people speed up their data analysis pipelines and so on. But the thing that always occurs to me is that can't we use that synthetic data to help us with decisions? And it may well be decisions about whether or not it's worth going and getting the real data or not. So that if you could access synthetic data that showed that there does appear to be some benefit of this treatment, we're not quite sure how large the benefit is, and therefore it might be worth going and sort of getting hold of the real data, or the converse, that we might sort of say that really there's nothing going on, and that we're probably better off spending resources, using resources in other areas. So I just raise that as a thought randomly.
Far from random, really helpful. I, for one, would see synthetic data and ISTs as being almost identical with respect to some of the issues we're talking about today, but others might have a different view. Mark.
Yeah, so I just wanted to add to that comment on synthetic data that one of the opportunities that we have is that, like, this is obvious, but rare things happen rarely. And if we're trying to uncover what's happening in the real world at the tail of these distributions, where we may have, on the one hand, really positive things happening, or on the other hand, really negative things happening that we want to gain insight on and uncover, the in silico world, or the development of synthetic data to explore or augment the data from the real world in those parts of the curve, are a great opportunity for us, especially if we're looking at the harm side, and it would be unethical for us to induce that harm in the real world, but we can fully explore it to the extent our tools allow. with the synthetic data world. So I think that this represents a significant opportunity for us to advance in this space. And I was curious if Parker had a thought on this, not to put her on the spot, but just coming from the Six Sigma world, I didn't know what her thoughts were on that.
No, for sure. Because I mean, those are the questions that are the most important. Right? You're always like, and you're like, how do I answer this question? How do I answer this question quickly? Right? And yeah, I think the use of synthetic data is really powerful, but you have to be extremely clear and careful in how you use it, right? For that, especially with talking to leadership and communication, et cetera, right? But yeah, I think there's huge potential in using synthetic data for the right question, right? To your point, and using it to augment is what we have is a huge opportunity as well. Don't know if that answered your question.
Thank you. Delia.
I just wanted to respond to Keith's point about the fact that there might be reasonable or understandable reasons for the difference or the discrepancy between the results. And that's exactly part of the whole idea about validating against RCTs. And what RCT duplicate was doing is actually shown where we can replicate the results and where we didn't, or we're not able to replicate the results, why we were not able to. So the whole approach helps with increasing trust and confidence in these types of data, and just understanding why it can align and why it doesn't align, and maybe there are legitimate reasons why it didn't align with the results of the RCT. So that it helps with understanding or getting that confidence in the data. And to Alex's point, it's about both sides. It's about the data, the quality of the data, its suitability and our trust in this data, and also in the modeling itself. So there will be standards for the modeling and how we can do it right, and best practice for modeling, and actually standards for the data and its suitability and its quality for answering the questions we want to answer using it.
Thank you. I'm keen, before I go to Martin, I'm not sure we've really nailed down with respect to the framework we've heard about today, something about the practicalities of these credibility weightings. I think that's an interesting thing, how that might work in practice. Marty, you might reflect on that now, or just start with what you're going to say anyway, and we'll come back to that.
Thank you. So I was going to raise a point because of the introducing the discussion on synthetic data. And I've tried to make this point very subtly in my talk. And I think there is a spectrum of integration of modeling and simulation and empirical data. So in my presentation, I purposefully chose Alex's example, which is a fully mechanistic model with some input data being used by imaging and description of anatomy and data on flow diversion, but it's mainly mechanistic, and the causality, the mechanism for causality is mechanistic. I think there is the other spectrum where you integrate modeling and simulation with empirical data and more data-driven approaches. There is a spectrum of integration of modeling and simulation. I think synthetic data is one example where you're integrating some modeling and simulation with causality that it's data-driven, empirically driven, which is, again, the other spectrum. And there is, again, a variety of ways in which you can use modeling and simulation. And that's why it's simpler for me at this point in time to think about the more mechanistic In-Silico studies, but there is that spectrum. And I think it's really important to consider the origin of the causality element to really think about what evidence of the causality we're actually dealing with, where they're fully mechanistic, where we have a explicit structural assumptions that we can judge their credibility, or whether more empirical evidence of causality that we're in some way integrating some modeling and simulation. I know this doesn't answer any questions, probably raises more questions, but I think it's a really important point that distinguishes this area and sets out this scope, this spectrum of approaches that makes it the complexity being exponentiated.
Thanks, Martha. And I guess when you're on that spectrum, as you move more towards the empirical based integration, that fades into what we're much more familiar with, at least in the HTA field, right? Keith.
Yeah, I mean, it leads on from what Martha's just said, but I also I wanted to pick up on something Dalia sort of said. There's another way in which in-silico trials could be potentially used. I was talking about explaining the discrepancies and exploring the discrepancies. But if you're in the situation where you've been able to replicate trial evidence using in-silico trials, that might open up an opportunity to sort of say what would happen in a broader patient population, so that you might, you know, I mean, well, we're probably all working on this, but you know, I'm working on how to generalize trial results to broader patient populations in terms of reweighting and so on. But you could think of it about, well, here I've got this model, I've got this in-silico trial. I've shown that you can replicate this trial that has been done, but now let's use it to actually speed up the decisions in broader patient populations for whom we haven't got trial evidence and we might never get trial evidence.
Absolutely. Alex, I'd like just to come in at some point in a minute on a Midden's question on the Q&A, but Mark, and perhaps you're going to answer it, but otherwise just go ahead.
Yeah, so on that comment about how to think about comparing in silico clinical trials with RCTs, there was some work that was done on this maybe 4 or 5 years ago, where it was a public-private partnership that had the FDA and several industry participants, and they worked out like a Bayesian method for evaluating the overlap between the results from a model compared to the results from a real-world clinical study. And based on the overlap between those two sources of evidence, then they treated the real-world study as the source of truth, and then you could weight the results from the model to kind of give you additional credit and potentially reduce the size of your real-world clinical trial. But again, that's based on the idea that your real-world study is the golden source of truth, or the ground truth in that approach. But there are methods like this out there.
Thank you. Before I go to Dalia, Alex, do you want to just chip in to answer Midden's question?
Yes, so I'm not sure if I'm really going to answer him, but what I would say is, Midden, the advantage here is that you can define how much you want in that regard, which you not always can in a real trial, because you have so many ethical, practical, constraints, access to patients, certain aspects you can't just simply do, that you never know actually what is the limit of risks of your products. You don't know, you haven't really stress test your designs to a large extent. And we recently found out with Mark Palmer a publication where it was said that over, on a 200 type of studies on complex diagnostic therapeutic devices, about 53% of all the class, the 53% of all the class 1 devices, which were about 40 something, within the top 5 reasons for root causes of failure, the top one in 53% of them was device design, was the engineering design. If that was if that was an aerospace or an automotive company, that would be bankrupt by now, right? But we still can do that. We can have it in medical technology simply because we are very limited in terms of what amount of physical testing we can do, which is not very different to what happens in aerospace, where you simply cannot test a lot for other reasons, right? So I would say the ability to do design exploration is an opportunity here. And I would say the limit is the limit of the amount of data potentially you have access to in some regards, and also the limit of your basic science knowledge that basically underpins the epistemic knowledge essentially that you have for building models in the first place, right? So I hope that helps a bit, Milan, to answer.
Thanks, Alex. Dalia.
I wanted to give a very similar example to what Mark was talking about, the framework for combining the evidence from the different sources from the clinical study or RCT and the modeling. There's another similar parallel also for a framework for combining evidence from RCTs and real-world evidence that SB or International Society of Pharmacoepidemiology have endorsed actually, which looks at how much weighting should we give to the observational evidence or real-world evidence compared to the RCT, and based on that, we are in one of three situations where is a low bar or high bar or a middle bar where you give equal weighting to both, or give higher weighting to the RCT evidence compared to the observational, or the opposite in the three. So there are frameworks that looks at how that type of weighting that you can use to decide on how you combine the evidence from both sides. And coming back on Keith's point about looking at how to use modeling and simulation data to estimate effects in a population will never be able to test in RCT. That's exactly the area where we are trying to consider in terms of testing how we can use modeling and simulation data to inform decision making. It's where we don't have the RCT evidence for these populations, elderly, the pediatrics, the pregnant women. Can we use modeling and simulation really to help us have the estimate of effect in these underrepresented or not represented at all populations in RCT data? So definitely this is an area where the use of this data would be highly valuable.
Thank you. Carl.
Sorry, yeah, I just kind of wanted to see if I could bring it back a little bit, because I guess at the core of what we were trying to present was conditional, believing that we've been able to reflect all sources of uncertainty, that we have a framework, a well-established framework that goes back 40, 50 years, where we can estimate the value of acquiring additional evidence. And that means that we can directly compare that to the value of providing access now. So it provides the natural way in which we can ask that question, when is there enough? We can also use it to incorporate questions about supporting innovation as well. So we can integrate all those considerations. That the key question, which I think we've been debating for most of this discussion, is how can we make sure we reflect all sources of uncertainty in our assessment? And that is a really difficult thing. And the more you think about it, you either become dishonest or you go insane. But I think what we were proposing was that when it comes to credibility, we shouldn't simply take the mechanistic causal relationships from In-Silico necessarily at face value, no matter how sophisticated and well-founded they are. Actually, everything's pretty damn complicated and we may have missed a bit. And for that reason, we should always compare them to the alternative. What would we save in the absence of In-Silico? But as we acquire sample information, we don't just update that prior, we actually change the credibility weights. Now, somebody was asking, well, what credibility weight should you put at the start? I have to say, I'm not sure about that, and I'm sure work's been done there. But it's going to be an interesting question about how quickly your initial credibility weights disappear as you sequentially sample more information. So I think we could, the process of updating not only our estimate of what the posterior looks like, but the credibility weights put on these two extreme alternatives means that maybe we're not that sensitive in terms of how much additional sample information we're going to need to our initial credibility weights. So yeah, that was kind of my comment. I was kind of trying to bring it back to the principles, what people think about the principles of value of information, given the difficulties that we do have about fully characterizing all the possible sources of uncertainty.
Thanks, Carl. Mark.
Yeah, along those same lines, I think that ties nicely back to Natalia's presentation, which raises that question of can we do an example where we can show the value to the system of injecting In-Silico into that framework? What is the economic benefit? How does it influence the way we estimate the or carry out the decision-making process, and really look at it from whether it's actually introducing financial benefit to the system or reducing harm to patients in the system? Like, and it seems like that's a framework that is there that we could capitalize on to quantify the effect and maybe show in an example.
Yeah, that's one for us to ponder, that's for sure. Marta. You're on mute, Marta.
The other element sorry, I just wanted to add to Carl and Mark's point. The other element is to recognize that the current kind of decision processes are very partitioned and very long. And to think that, for example, in the case where you have a technology that might be appropriate for more than one population, for example, the cost and the time of these partition processes mean that to reach your final indication, it might take you years until you can sequentially producing RCT evidence for all the indications you might need to. And I think one of the potential values of In-Silico evidence is actually to prioritize the collection of evidence to populations where you might have more value of collecting that evidence. So I think there's a question, I think just maybe going back to Joyce's point of systems and decision-making processes, I think there's a huge potential for this information for In-Silico information to improve and make more efficient the kind of existing partitions, regulation and funding decision processes, and linking very clearly to data collection.
Thanks, Marta. Dalia.
This will be a very useful use again for this type of data, Marta, and it would be great to explore it, but also to agree that this underlines or underscores the importance of that thinking from the whole life cycle, if you like, of the technology and the evidence generation process, and not like steps. It is a continuum, as Carl is saying, and updating our knowledge based on the newly collected data and updating that, and maybe that's an untapped area for better use of value of information. And also I know that I'm personally, and this is again personal opinion, is that I feel like it's underused. And some HTA agencies have started, like Zen in their latest update, the Dutch HTA agency, now require value of information analysis for all submissions. So there is value of thinking about value of information analysis and how we can make better use of it. On the other hand, we need to think again that any new type of approach or data that we collect, or type of data like modeling and simulation, will come with its own specific uncertainties. So yeah, we think that this evidence will help us solve some uncertainties, but it comes with its own different types of uncertainty that we have to be aware of, to just to caution. And again, this might be another way of thinking about where can we better use value of information and these like living evidence generation approaches, or optimized evidence generation approaches. And thinking out loud here about living HTA, for example, some topics will be amenable to this type of thinking, where we can use that continuous evidence generation and know when to stop, like collecting further evidence or generating or doing further research using both value of information. But other approaches that we thought about, like sequential trial analysis, have been mentioned as well as a potential approach that could help guide whether further trials are needed in certain areas. So I think these approaches are underused, and maybe that's the research community, the academic community, that need to invest a bit more in terms of like making it more visible how they can be useful for decision making, to convince HTA agencies and regulators to work together to make better use of these approaches.
Thank you very much, Julie. We're 4 minutes or so away from completion. What I'd just like to do, just for the final couple of minutes, Alex, if I may, is to ask you about sort of next steps, as it were, in this project. Marta, if you want to chip in as well, specifically in the area of next steps relating to the specific material we've talked about today. Alex, do you want to say anything on that to start?
Yes. So as I explained at the beginning, we are in a phase of discovery, which means events like this that can generate new items, new leads of things that either confirms what we are thinking or gives us different perspectives to consider gaps that we have in our thinking. I would say if you're interested in general to this particular area of decision making, Marta is your lady. She's the person who is leading in our discovery phase, together with Mark, and I'm providing overall leadership because there is about 9 discovery streams. This is one of a number of them. So you can reach out to me if you have general interest in knowing more about the network and what we are developing. I put quite a bit of material already on the chat. Hopefully you've seen that. And specifically, I would like to invite you to contact Marta, connect with her, and also with myself more broadly in other areas. Please try to join 2 things that I put on the chat: the credibility assessment workshop that is in the coming days, where you will get practical insights about what it is. And I think we believe that part of the building trust and credibility is about understanding. Parker put it very nicely, is the ability to learn. And you cannot learn if you don't understand and you have some basic. It's like learning a new language where you don't even have any clue even on the grammar, right? And you can hear things, but they don't consolidate. And this is what we're trying to do with this discovery phase, is help with some foundations so that as we all hear things, we all connect the dots and can engage. And then I also put in the chat something that maybe has gone a bit unnoticed, which is a survey. Can you please help us to complete that? This is a capability mapping. So another stream is looking into what skills will be required for the next generation regulatory affairs people, and also what sort of skills around modeling and simulation in regulatory decision making, but also other areas of decision making will be critical. And we want to have as broad, canvas of opinions as possible. And we will, of course, share in our Zenodo community space all the findings that we are doing through reports as soon as we sort of consolidate them. And I know that there are ideas from Marta as well on putting some of the key findings that we are gathering around this particular domain as well in some sort of white paper, some sort of written format. So with that, I just want to say a big thanks to the crowd in York, Marta and Mark and Carl as well, and Natalia, for all the work you put in putting it together, and for all of our panelists as well on behalf of the network. I'll hand over for Marta to close it.
Thank you, Alex and Mark. Yes, I think as Alex mentioned, I think the next step for us is to put this down on paper. I think the webinar and discussion today was extremely helpful and positive. And I think it was a really good experience in kind of merging a number of disciplines represented here today. And our chair, Mark, had a great effort in kind of joining up these 2 or more kind of different people with different expertises. And I think we managed to get a common language going, which I think is really difficult to do when thinking about different scientific areas. And I think obviously because I think the In-Silico is quite aligned, as I tried to highlight in my talk, with our kind of decision-making and decision analysis framework. I think it was quite a smooth process, and I think everyone potentially will agree with me. But I think getting creative, using Joyce's words, but getting creative on finding solutions is the next step for kind of this innovation, for in-silico clinical trials, on how they can be efficiently and appropriately used in decision making. So I'm hoping that starting with kind of small steps, and this workshop being the first of them, will kind of help us reach step innovation in terms of processes and principles. So yeah, thank you everyone for incentivizing the discussion today.
Thank you. Well, if there's only one thing I've done right today, it's finishing on time. Good for that. Thank you, everyone, the panel for joining, all attendees for contributing. Really grateful, and let's all keep in touch.